Thank you, Mike, for this very interesting talk. As for the other lectures, the questions will be after for the sake of keeping check on time. So I've been already introduced nicely by Francisca this morning. I am Robin Ménage. I just joined the Boehringer Villemi Clinic recently. And today I will talk about the gut microbiome with you. And I will explain you what are the changes in the gut microbiome and how they can relate to the effects of fasting on your health. You have to understand that as a human person, you are not just a person, you are an ecosystem. You are a person with your bacterial ecosystem, and we have actually more microorganisms in our bodies than human cells. This is a very nice illustration. This is a piece of art showing that we have a diversity of microorganisms even in our face. In this piece of art, the artist Melissa Fischer took a print of her face and seeded the bacteria which are on her face to cultivate them. And these are bacteria which are safely dwelling on our face, and this is part of our ecosystem. Moving more into the gut, the ecosystem in the gut is the more complex. This is where we have the most bacteria. And we always think about the gut microbiome as what we see in fecal sample. And this is just one tip of the iceberg, because actually our gut microbiome is composed of different layers, and you have some microbes which are just floating in our gut, and they are incubating, changing depending on the food we eat. And you have other microbes which are more attached to the gut, and they will snack on the mucus, which is here to protect our gut and absorb the nutrients. These are more stable, and when you do a microbiome analysis, these are more difficult to analyze. The gut microbiome, this is not just bacteria, this is also other organisms, viruses, fungi. We have a lot of viruses that you can see here. These are viruses which are attacking a bacteria, because these viruses are predators of bacteria. They attack the bacteria, and they are known as bacteriophage because they eat the bacteria. And these are very important also for our health. We just start to understand their importance, and it seems like the attack of these viruses on bacteria can also have an influence on the development of a disease like inflammatory bowel disease. This is an interesting picture because we don't see every day a gut microbiome, and this is the closest picture we can have. This is actually a fecal sample, so much pleasant to look at the microscopic level than at the macroscopic level. And you can see the diversity. All these small dots are bacteria. We have very small bacteria, very huge like this one, and this is really actually like an ecosystem, like a forest. You have a variety of organisms working together to survive, and they are also working with our human body. These microbes, they are called also commensals. This is meaning that we eat at the same table, so we are together, both our microbes and us. I will talk now about what this microbiome can do for our health. What you need to know first is that the microbiome is both unstable and stable. Stable because generally it is seeded at birth, and we keep the same fingerprint of microbiome for most of our life, and this is very personalized. Also unstable because this is depending hugely on the food you eat. This is what you can see here from the profile of someone who lived abroad for a year and who had his microbiome tested for a regular period of time. You can see that each line is one type of bacteria, And you can see that the changes are massive, and to get an accurate picture of a microbiome, you need to have really regular sample and an average of that. It depends a lot on what you eat. This is what you can see here. When you switch from an animal-based diet to a plant-based diet, you favor some types of bacteria and some others. Because when you eat meat, you will have secretion of bile in your gut to deal with fatty acids, and this can select some microbes, kill them, and others will be able to grow peacefully. The changes are really profound, and what happens in your gut doesn't have only an influence on your gut, but it can also affect distant organs like the brain, cardiovascular system. Pretty much every physiological function is to an extent depending on the gut microbiome. It has important effects on our energy metabolism. And this is what we can see when we compare obese individual versus lean individual. They have very different gut microbiomes, and this particular gut microbiome changes their energy harvest property. It means that the gut microbiome can extract more energy from the food and actually make you available more calories, and you can have 100, 200 more calories depending on the gut microbiome types, just because it digests it differently. This is what was shown beautifully in some experiments which was done at King's College, where we study twins, and there's some twins who are not exactly the same. One is lean, one is obese, and wanted to know if taking the microbiome in isolation, you can influence how the obesity phenotype will change. And this is what was shown here with these mice, because when you take the microbiome of an obese twin and you put it in a mouse, the mouse will have increased adiposity and will get fat. When it's coming from a lean individual, the mice will be staying lean. It's modulating energy metabolism, and it's not just in case of disease, it has very physiological functions. If we have these bacteria in our gut, it's in most part because they have been selected by evolution. And we start to understand why, because when you compare the microbiome of bears between different seasons, between a season where energy is available and the bear will feed a lot to store energy for the winter, you can see that the two microbiomes are very different. And in fact, the fact that a microbiome can extract more energy was really useful in the evolution of these animals, because it means that they could extract more energy to prepare for the winter. So this is why, in some cases, you have microbiomes which can take up more energy, and this can increase adiposity in individuals bearing these microbiomes. I can't talk about hibernators without mentioning this very interesting study, because we are also interested about muscular mass and its changing during fasting. And in this study, it was shown that even in the squirrels, the gut microbiome also helps not just for energy metabolism, but also to preserve the muscular mass, because it was shown that the urea in these animals, which is normally a byproduct, a waste, which is excreted, it was used by the microbiome to make up new amino acids, and this allowed the preservation of muscular mass. I will now talk about the changes in the microbiota during fasting, but I will start by the changes in the gut structure first, because for the gut, fasting is not just an energy crisis. This is also a housing crisis for the microbiomes, because the gut is changing a lot during fasting. This is what you can see on these pictures which are electron microscopy image taken by some collaborators from CNRS in France. You can see on the left the microbiome, the gut of an animal during the fed state, and on the right, the gut of an animal during the fasting state. And what you can see is that the profiles are very different. The gut is becoming very smooth because absorption is decreasing, and we start to understand what happens in this case, which is corresponding to regeneration of the tissue. During this stage, the mucosal mass in the gut is decreasing a lot, and the volume of the organ is decreasing as a consequence. This is, at the molecular level, explained by change in the cell cycle, which is slowing down, so the proliferation is slowing down. At the same time, you have an increased rate of apoptosis and autophagy, which is recycling your old cells. This is a bit like circular economy for your body. You're recycling the old damaged material to make new one. And this is contributing to the decreased intestine volume during fasting. What we know is that there's also a stem cell activation leading to regeneration of the intestine. At the molecular level, this is mostly driven by some nutrient sensing at a pathway which is called mTOR, which is very important to controlling balance between proliferation and cell death during feeding state and fasting state. This is what you can see on some of these complicated slides, so I will not go into the detail, but you can look at this paper if you want to understand it better, because this was written by the godfather of mTOR, David Sabatini, who had actually a lot of problems, but he was in his way to almost get a Nobel Prize, but unfortunately there was some problems of personal problems, let's say. Now, let's go on about the gut microbiome during fasting. So I will talk about the study we did at the clinic in 2016, where we evaluated how the gut microbiome and other parameters of the gut were changing during fasting. So we looked at the bacterial composition, but other markers like inflammation, gut permeability, and energy metabolism to understand how this is changing as an ecosystem as a whole. We took some samples before and after the fast, but we also took some sample after 3 months after the fast, so we could understand if the effects are persisting. We could document the usual change in energy metabolism during fasting at these 4 time points: before the fast, after the food reintroduction, and after 3 months. And we can see that there is this classic metabolic switch when glucose is not used anymore as a source of energy for most of your cells, and most of the energy is coming from fat burning. We did some sequencing of short fragments of DNA in the gut. This is actually the most common method to identify microorganisms because they have different DNA, and if you sequence this gene called 16S, you can figure out what they are. We managed to get insights on quite a lot of microorganisms. Most of these are bacteria because this gene is not present in viruses, and this is a different one in fungi, so we only study bacteria here. There was a decrease in some specific microbial species, and these were the ones which are using plants as a source of energy. So this is kind of logical, but this still was new that when you stop eating, the bacteria which are using your diet for survival, they are inhibited and they can disappear. This is what you can see here at endpoint 2 and 3. These are almost undetectable. And what's very interesting is what I told you earlier about hibernators. When we compare the effect of fasting to what happens in the wild, we can see that the responses are very common, and the microbiome of a squirrel which is hibernating is very similar to the microbiome of someone who is fasting. I always take this example, and it's quite cute, so I took another one today with this bear, because actually the microbiome of bears which are hibernating is also very similar to the microbiome of people fasting. And this is showing that the bacteria which use the cell fragments, the cells which are recycled during fasting, the rest of your intestine regenerating, these are used by bacteria for survival, and these increase in abundance. As you can see here, the effects are still found during the food reintroduction, but not anymore after 3 months. You can see that the effects are very similar because this population of bacteria, the Bacteroides, were the ones which were the most affected in our study, but also the most affected in these animals when they were hibernating. At the gut permeability, because this is very important, when gut permeability is altered, you can have a leakage of some bacterial compound in your blood, and this can be a source of inflammation. We found that the level of the markers of gut permeability in our study were quite stable across the different phases of the study, and this was not expected because some studies in animals were suggesting some change, but we found that the intestinal barrier was not compromised and was staying the same. We also measured the level of different inflammatory markers, which were really informative because we showed that these markers were suggesting that there was an immune reaction associated with the food reintroduction. This is very interesting because fasting has clear anti-inflammatory effect. We know that because this is a really successful therapy for inflammatory disease, neuroinflammation. And this finding suggests that it's not only fasting which is important, it's also the period of food reintroduction. And it can be as important as fasting itself because when the bacteria which are depleted in your gut are refed, if they are refed with some pro-inflammatory diets, you will get an exacerbation of inflammation, and this might be detrimental. So this is why the period of food reintroduction is very important, and it's very important to follow specific guidelines for keeping the effects of fasting as long as we can. We also measured these consequences of changes in gut permeability, so the bacterial lipopolysaccharides, which are markers associated with different chronic disease. And we found that one of these products was decreased during fasting and remained reduced after the food reintroduction, which means that we are less exposed to bacterial toxins, and this is favoring a more resilient and regenerative ecosystem during fasting. We also found that some very interesting markers of inflammation, like the secretory IgA, which are good reflect of protective action on the gut, they were increased during the study, which shows that there was an immune reaction, but this immune reaction was creating a situation where there was an adaptive response, and this is increasing immunogenic potential in the gut. So I told you that the changes in gut microbiome were coming back to normal after the fast, and this is in a way good because we also want proof of safety. What we also found is that even if the gut microbiome composition was coming back to normal, we could still see some benefits looking at the function of the gut microbiome. And this is what we found looking at short-chain fatty acids, which are product of fermentation of fibers and which are known for having signaling effects leading to protective effects on various inflammatory diseases. And these, as you can see on this figure, they were increased at the time point 4, which is the time point 3 months after the fast. So very interesting, suggesting that the effect can be persistent in a way. The changes in the gut microbiome were also linked to energy metabolism. So we keep with the idea that what happened in the gut can also influence your energy metabolism. We could not make much conclusion about that because of the size of our population, and we need more sample for this type of studies. But there was interesting follow-up studies after our first study was published, and this study in particular, which looked at the gut microbiome and it's linked to blood pressure and body weight during fasting and during a different intervention, showed that effects were very similar between this study and our study, suggesting that what happened in our study is not just specific case, but can be generalized. And what was interesting with this study is that they show that the effect of fasting on blood pressure was persistent and was found 3 months after the intervention, but not in everyone. It was found in people with specific gut microbiome phenotype. And so it was really interesting to find out that baseline microbiome, so before the fast, can be used to predict the changes in blood pressure 3 months after fasting. This model was developed on their own data set, but it was also working on our data set, meaning that this has the potential of moving into personalized nutrition strategies for fasting in the future. I will mention briefly other technologies which can be used to evaluate the gut microbiome, because what we did was based on short fragments of DNA. This was the tip of the iceberg. Now, with decreased cost of microbiome sequencing, we can have access to better machines which can do sequencing in real time, more deep, at a better cost. Some of these technologies, I used them when I was still working in King's College to evaluate the effect of pesticides. Here it was effect of pesticide on bumblebees. And using this machine was really phenomenal because injecting the sample, after 10 minutes, I could see the first profile, and it gave me a profile of the gut microbiome of bumblebee within minutes after the injection of the sample. So this is really a revolution in DNA sequencing that you can have real time. What's important is not just what are the microbes, but also what they are doing. And for this, you can use different technologies. One of them, we heard this name quite a lot already today. It's called metabolomics. We didn't do it on fasting, but I wanted to take an example of a study I did on also pesticides. It was in the Twins UK cohort. And you can see that these technologies can be used to profile thousands of metabolites of different sorts, not just amino acids, lipids, and different of sugar, but you can also have access to the exposome, so what we are exposed to. So you can see also what drugs, pollutants we are exposed to. And this is really interesting because you have an incredible level of detail. Here I just take an example of the polyamine metabolism, which is very interesting for some researchers doing studies on longevity because of these spermidine compounds which are attributed to have some longevity effect. I will also mention now the oral microbiome, because what happened in the gut is very interesting, but we are trying to move forward and look beyond, because microbes are not just in our colon. This is where we have the most microbes, but they are also found in other parts of the digestive tract, and in particular in the oral cavity where you have also a lot of microbes. Some of them are pathogens, but some of them are just commensal, like in the gut. They are here and live with us in peace. But some of these microbes can have pathological effects because they can be associated to some disease like periodontitis, and having a microbiome profile in the oral cavity is more and more used to guide intervention in dentists. It's also involved in other pathologies like diabetes, pancreatic cancer, and this is very surprising, but in a way, the microbiome in your mouth is also very close to your blood circulation. In the gut, you have many layers protecting from bacterial invasion, and in the case of the mouth, the layers are very thin, so you can have an easy translocation of the bacteria into your bloodstream. And we've been really interested about that because one of the symptoms which occur a lot during fasting in the patient at the clinic was the bad breath. You can see here, this was the symptoms which were reported in the study published in 2019 about the effects of fasting in 1,400 people. And this bad breath, we know that this is not necessarily the smell of ketone because this is very different, but this is due to some pathogen in the oral microbiome, and actually there's no study showing whether fasting can change this population. So we are doing our own study, and this is ongoing. We will start the recruitment soon, but we will do the first study of the microbiome both in oral cavity and in the gut. These are reflections for other study because there's actually a lot of points which can deserve to be explored in other studies. We need to understand how the enema can have an influence on the change in this population, because this is a regular, normal practice during Boehringer-Wilhelmy fast. You cleanse the gut, and this is generally improving the well-being of the patients, but we don't know what it does actually on the microbiome. And we need to understand if the changes we can see during the fast can be attributed to the diet or can be attributed to these also practiced during the Boehringer-Wilhelmy fast. We need to understand also if some effects of a Boehringer-Wilhelmy fast can be linked to the gut microbiome, like the mood enhancement. We know that the gut microbiome is very important for the mood. It can be secreting some hormone, serotonin, dopamine. They have influences. They are influenced by the gut bacteria. So it will be very important to understand if the gut microbiome can influence that as well. What about the viruses and the fungi? We know that they are very important, so this is an obvious exploration for future study. We also need to understand if there is what we call a decreased colonization resistance. If during fasting, the fact that the bacteria are depleted, is it leaving the opportunity for pathogens to grow? There are some studies which suggest that it is the case, but it is the case when you feed these pathogens with protein. So some particular diets, and this has been published last year, but this is not necessarily generalizable, and we need to understand what happens exactly to this pathogen during Boehringer-Wilhelmy fast. We need also to understand the link with the lipid metabolism remodeling, because the gut microbiome has an influence and can control the cholesterol level. We need to understand also the effect of probiotics, because this is a very common practice to give probiotics during the food reintroduction phase and sometimes during the fast. But I know that the community is really divided for that, because some people think that giving probiotics will have some benefits, promoting the colonization of your gut by good bacteria. But some other people think that giving probiotics will delay the colonization by your own microbiome because these bacteria from the probiotics, they will take an unnecessary space, basically. I will finish by some recommendation and some pitfalls for clinicians because I can see more and more that the gut microbiome is profiled by some companies and that they provide reports saying that people are healthy or not, that this is normal or abnormal. And this is still an emerging field of research, so most of these are not ready for translation into the clinic. And some of these pitfalls are that we should not expect that an individual's microbiome will be stable. This is what I showed you. It depends on what you eat. It depends on where you live. And so one analysis of the microbiome is like a snapshot. You take a picture and you look what it is. But it doesn't mean that the day before or the week before it was the same. And not everyone will react the same to a diet change. Everyone has its own microbiome. I have some studies with collaborators at King's College. We show that people eating blueberries, some of them have effects on their blood pressure because they have some particular microbes in their gut snacking on the polyphenol and having some good effects. And some people will not have this effect. And there is no standard microbiome ecology for healthy people. It means that it's really difficult to say that a microbiome is healthy or not. When you compare different profiles, we know that in general, bacteria which are from the firmicutes, for instance, these are good for your health because most of them can use fiber as a source of energy and they will have positive effect. But this is not necessarily always true, and you have some people who have very, very low level of these bacteria and which are perfectly healthy. Different strains of bacteria can have different effects. It's really interesting when you see a bacteria, a name that you probably know, E. coli. This is a bacteria which is responsible for food intoxication. But some strains of E. coli are actually very important probiotics, having some positive effect for health, and they can be involved also in the synthesis of vitamin, for instance. Different laboratory and computational methods will not always give the same result. This is perhaps a bit surprising, but this is an evolving field of research. So the gene catalogs, microbial catalogs are always completed, and a study which has been done 5 years ago, if we reanalyze the raw data these days, it will give different results. Different results, I mean, not necessarily opposite conclusion, but maybe revealing something else, something new. Another and last aspect which is very important is that the reference range, which is used for blood tests, this is not necessarily working for the microbiome, and this is for the reason I described above, and so that's how you can conclude that if you look at someone and it has some specific number of some bacteria, it's really difficult to say if it's healthy or not. And this reference range, sometimes it can be established, but it has to be established on a specific population with a specific method, and all of this has to be the same when you analyze the actual sample. So it means that the microbiome tests which are sold, it's not necessarily wrong or a scam, but we have to be careful with interpretation and always look at our own population with our own reference value. With this, I thank you for your attention, and we'll be happy to take some questions.